So welcome to our distinguished panel and all our participants. According to the World Food Programme in its 2020 report on the state of food security and nutrition in the world is not on track to achieve zero hunger by 2030. It was estimated before the COVID pandemic that the number of people affected by hunger will surpass 840 million by 2030, almost 10% of the world's population. The world's population is projected to reach 9.1 billion by 2050, and the Food and Agricultural Organization of the United Nations, or the FAO, estimates that global food production would have to increase by 70% to match this growth. To this end, we would need to increase land productivity and conserve natural resources in the face of a multitude of severe challenges, which include climate change, increased droughts, flooding, and rising sea levels, which hamper crop productivity and make farming a high-risk venture. Nuclear security and technologies are part of the cutting-edge research and development efforts that are accelerating globally to develop and implement sustainable, climate-smart agricultural practices. These technologies not only increase agricultural productivity, food security, and nutrition quality, but also improve the livelihood of farmers. Importantly, these technologies benefit instead of deplete the natural resource base that sustains these agricultural practices. Today, we have the opportunity to hear about the contribution of innovative nuclear technology to sustainable agriculture development from our panel of experts who come from every corner of the globe.
So, okay, let me start now. And indeed, again, thanks for the opportunity to say a few words about useful or peaceful applications of nuclear technology, specifically then for improving crops. The topic of my talk, as mentioned, is I'll be setting the scene, kind of explaining the technology, and also briefly explain a bit the role of the IAEA in I would say, promoting and doing research using mutation breeding. So if I can have the next slide. So here's the outline. I'll just be briefly explaining what crop irradiation breeding is all about, the technology, the type of support that the IAEA is offering to its member states, then some explanation also on the advantages and the challenges where we are with mutation breeding, and then what the prospects are of radiation-assisted crop improvement. Now, before moving on, the terminology that is used most typically is mutation breeding. And as already mentioned, there's actually, it covers both physical means, so nuclear technology, and also chemicals. Keeping with the topic of today, I'm going to use more the terminology irradiation breeding, so I will not be talking about chemical mutagens. So next slide, please. Here, I guess I understand that there's a mixed audience out there. We've seen that, so I'll be talking about. Here's the first slide on explaining what mutations are all about. So you have spontaneous mutations and you have induced mutations, and a mutation is nothing more than a change in the genetic makeup of an organism that is heritable. So it goes from one generation to the next. And it's something that happens naturally in all living organisms. So that's very important to understand, that this is something that happens over long periods of time. And I've shown here a couple of examples in barley, different types of ears that you can have, seed, number of seeds, a lot of variability that mutations are creating spontaneously in nature. And the other example here is just the color of maize kernels that you see, so a wide variety. So mutations are a very important driver of diversity. Now, coming to induced mutations, that means you can actually use techniques such as irradiation to generate similar mutations, except that you do it much more rapidly. And some of these mutations create variation that is useful for plant breeding. You see here a schematic, a simple drawing of DNA, and you can have different types of mutations. I'm not going to go into the details, but the different types of mutations have relevance on how they can be applied. Next slide, please. Here, I'm just shortlisting the types of sources of irradiation that we have at the IAEA, specifically in the laboratory in the campus close by here in Cyprus, Austria. We have two types of sources. One is a gamma ray radiators. We actually have two different types. Here you see one of them. Obviously, the gamma ray irradiator has a radioactive source that is cobalt in this case, and that decays over time. I've indicated here the half-life of the cobalt source that we have is called the gamma cell, and that's 5.27 years. That means it decays over time. Very important here for applications is the dose rates of these sources of radiation. In this case, in case of gamma, is actually the dose rate of the machine at this point in time that we have is about between 50 and 60 gray per minute. That's gray per minute. That just means it's a measure for how much radiation is administered to the in this case the plant material. Below you see a non-radioactive source of radiation. So you can also have non-radioactive sources of irradiation, and this is an example of an X-ray machine source. You'll be hearing more about that by some of the members in the panel. But this is what we have in the campus here. We have two models. One is a model. These are retrofitted. These are all for research use, really, or for applications in the medical sector. The one that we mostly use is the RS2400, and that one has a dose rate which is much lower than the gamma ray. This is about 9 grays per minute. This is very important when it comes to the applications for mutation breeding. On the right, you see the ray cell, MK2. That's something that was acquired more recently. This is actually a blood irradiator that we use for applications in food and agriculture. And in this case, the dose rate, very important, is 6 grays per minute. So there's a difference between the gamma sources and the X-ray sources in terms of their dose rate possibilities. Next slide, please. Here you see the types of plant materials that we can actually bring into these machines and that we can, where we induce variations. And you can see there's a whole lot of different types of materials that we can use. You have seed, you can use whole plants if your container is large enough. You can also have various parts of the plants. You can have tissues, in vitro tissues. You can even have single cells. You can also have pollen. So there's a wide variability in plant materials that you can subject to these sources of radiation. What's important here to say is that depending on the type of material, you may have to increase or decrease the dose of radiation that you administer. So in terms of applications for mutation breeding using X-ray or gamma ray, it's very important to know the dose. So we actually need to be able to have a range of doses. I would say typically using X-ray or gamma ray for most of the plants, they would go in between, let's say, 5 to 10 grays up to 2,000 grays. So that's very important to remember. Next slide, please. This is just an example that shows how it is done in practice. Basically, you have two steps. The first step is the mutation induction, and I've written here, this takes really seconds, 2 minutes, sometimes up to an hour if it's an X-ray, but basically, it's very, very fast. So, and this is the step where you induce all the genetic variability in the plant material that you are using. If it is seed, then it will be in the seed. If it is single cells or cell cultures, then you will induce the variability in these individual cells. So you will have typically bags of seeds. In case of a seed crop, you see here on the right, in case of the gamma cell, where the material is positioned, and then it is exposed, and you can determine that beforehand how long you want to expose this material to the source of radiation. In this case, it's a cobalt-60 source. And then typically, when you're starting with a new variety or a new plant material, you will subject the material, and this is illustrated here, between the dose ranges. So this is an empirical test. It's very quick, but it allows you to estimate how much radiation you have to administer in order to have an effect. Here on the left, you see this is the control, no radiation given. On the right, the extreme right, you see here you've administered 600 gray, everything is died. And then we know from experience that somewhere halfway Here at this dose, somewhere 200, 300 in this case, you will have induced sufficient variability to now bring it to your breeding program. And you can plot this on a curve. Then the next step is, once you've done this on a small scale, then you can take a large bag of seeds, and you can then subject it to the optimal doses, usually 2 or 3 of these doses. Then I can have the next slide. Then we're moving to the selection of the mutants. Again, this is an example of a seed crop that is a sulfur. As mentioned, it depends really on the type of material that you use, but roughly you will start with a seed. You will irradiate it. It's also important to say, I often get the question, or we often get the question, once you irradiate the seed, for example, the seed itself does not become a source of radiation. It just looks as before. It's like, you know, somebody's going to have a dental check. You get an X-ray. You know, once you leave the dentist, you yourself don't become a source of radiation. So it's very important to say there's no transfer of radioactive material or cobalt from the machine to actually the seed. So, but then we have to go through a couple of cycles, and this is what is the time-consuming aspect of it. So you will get from what we call an M1 seed, then you have to grow an M1 plant, and it's in subsequent generations then that you will start with the selection. Next slide, please. So that's also very important to say. The types of the radiation induces random variability. So these changes that are occurring, that are induced, are randomly distributed throughout the entire genome. So that has implications, of course, on your chances to pick up a useful variant, because the great majority, of course, of the variation that you are inducing will not have any value, any practical value for plant breeding. So the trick really is that out of the many variations and plants and mutants that you are generating, is to find the one that is useful for plant breeding, and that's where the bulk of the work is taking place. So, and this is a bit summarized here: mutant line development. So this takes years, not minutes or seconds or hours. And here you have a couple of different strategies that national programs, breeding programs, mutation breeding programs, I'm sure you'll hear more about in the next speakers, that you can take. If your mutant is perfect, in a sense that you have the new trait and all the other characteristics are preserved, then you can go straight and have a very quick mechanism here to release here, sorry, on the right in the red, through rounds of selping, and you have a variety. That's not always the case. It could be that you've introduced several changes, some of them good, or maybe one of them good, but some of them are not appropriate. In that case, you can actually try to move the useful trait into other germplasm through process. And that also takes time. And there are technologies available, biotechnologies, molecular tools that you can use to speed up this progress. And that's basically what you see here highlighted in yellow and here also in blue. I don't think I need to say more about that, other than there's different options to move either the entire plant, or the new trait that is of interest to you. Next slide, please. Yes. So here you see it, and that's what I said. You look at the seed, M0 seed, it's called M0 prior to the irradiation treatment. M1 is after the irradiation treatment. It's identical. Then you bring it to, for example, a field, and then you will go, in case of a seed crop, you will take it to successive cycles of selfing to create homozygosity. And of course, this is where you have, this is what takes the time. And then you have, you know, phenotyping, that's basically screening for the trait you're interested in using just various types of tools. Genotyping refers to a similar screening methodology, but now using molecular tools. And then you see here on the left, this is your mutant. This is an example from the lab, actually, where you see, you know, the amount of labor and work it goes if you want to preserve all these mutant plants. Next slide, please. Okay, a few words about you know, X-ray versus gamma rays. Generally speaking, at least my opinion, that there's not a whole lot of information out there that really have looked very deeply into the differences between the various types of radiation. There's been some work done in the laboratory, and here you see comparing X-ray versus gamma rays for a seed crop. And here you see again the two sources, different sources. Here you see a dose response curve, you can say, or radiosensitivity testing as we call it. Here you see the schematic, and basically you see that this looks pretty similar, in fact. So based on the experiments within the lab, you see that actually, you know, the types of changes, the phenotypic changes, the biological effect looks pretty similar. Next slide, please. And this is again just basically showing the same thing. And I have to acknowledge here the work of the plant breeder in the laboratory. This is what has been done by Dr. Ghanim. And again, here you see with wheat, barley, gamma X-ray, it's pretty similar when you look at it phenotypically. Next slide, please. Just touching on the impact, and these are examples from, I would say, almost historical examples of gamma ray-induced rice mutants. The IAEA actually has a database. You can check that, and this database gives a summary of all the, not all, but released varieties that have been documented over time, and I think about up to 70% of the released varieties documented in that database are produced using gamma rays. And here you see a couple of, yes?
Dr. Ingelbrecht, sorry, we need you to wrap up. So if you could move perhaps to what the contribution of the IAEA is, that would be great.
Okay, good. Next slide, please. Here is the next slide. So what is it, the support that IAEA gives to member states? Basically, there's financial support, we have technical cooperation projects. At the moment, these can be national or regional. So we have 53 national projects, and then there's 11 regional projects. And what do they do? What do they support? They support capacity development. That means human resource development. Also, infrastructure improvements. Of course, in the context of these projects, you can develop, the member state can develop and release new varieties, and typically they take 2 to 5 years, and they can be extended. And there's a technical officer throughout this project who can provide technical backstopping. So that's more in the area for technology transfer and applications. Then we have coordinated research projects. These are really geared towards developing R&D protocols and also mutant lines. And they're focused on global crop improvement challenges. So we look at what's an important challenges for multiple member states. Can mutation breeding help find a solution? And then that's how the project is being established. This is a 5-year project, and it's led by the IAEA technical officer. In the laboratory, we also provide crop irradiation services using either X-rays or gamma rays. So any member state, any out of the 170 plus member states of the FAO or IAEA can come right to us, send us their samples, and we'll irradiate that for them for free and return it back. Here you see the numbers for 2019, where we treated over 200 accessions from 42 different plant species, either using gamma or X-ray. And there's also a mutation breeding network that's being established, trying to bring different member states together to exchange experiences and resources. This is starting in Asia Pacific, and at the moment counts 15 countries. Next slide, please. I'm almost through. I think I can, have 2 more slides. This is just to wrap up the advantages and the challenges of radiation breeding. I think on the positive side, you see that this is a conventional breeding product. That means there are no special regulatory or biosafety requirements. So it's just a conventional breeding product like crossbreeding. It means also that this is accessible to the end user. And it's been around for quite some time. So there's a track record of safety for food, feed, and the environment. You'll hear more about that later. Also, it's a technology that is applicable to many crops and traits, I would say specifically the seed crops. Then on the negative side, I mentioned already that's a random mutagenesis technique, so you need to really look hard for the trait you're interested in or the crop. It's not as far advanced in case of the vegetative crops. If you have what we call polygenic traits, these are more challenging to improve. And then, of course, it requires a large scale. So sometimes if it fails, you're not sure why. Was your population too small, or didn't you use the right dose rate? So there's that possibility. And then, of course, there's also the concern about gamma ray, especially, and generally the use of radioactive sources, about sustainability, safety, security, and also a cost concern. And then, well, as I mentioned, there's actually limited knowledge on what these different types of radiation does at the molecular level. And that really limits the applications. Next slide, please. And I think that's my last slide, just to wrap up. Yes, there's tremendous potential if you combine the classical mutation technology that I just explained, that has been used for the past 40, 50 years, with advances in science and technology, including new sources of radiation. And I'm just, giving some examples here. There are advances now in cell and tissue culture that allow you to treat single cells, and this is especially relevant for vegetative crops. There's a wide number of new methods now for precision phenotyping at lab, greenhouse, and field level, so you can increase the precision and the throughput. Next slide, and then here of course the use of what we call big data and genomics can certainly increase the precision, accelerate the breeding. Here again, an example from the plant breeder and the molecular geneticists in the laboratory. You see here a tall plant, a range of semi-dwarfs. We've been able to identify where the mutation is located, and that can help you then to develop markers and move the trait more quickly. Another example from the plant breeding program in the laboratory is what we call speed breeding technologies. So you can actually accelerate, have multiple cycles of selfing per year, and that all of these things then reduce the work. Next slide, please. And I think this is my final slide. Yeah, just to concluding remarks. Radiation breeding is an established technology for increasing agrobiodiversity and for crop improvement. Very important. It's not technical, but it's very relevant. It is in the public domain. It's widely accessible to everybody, all member states. Numerous products have been adopted by farmers globally and are consumed on a daily basis. Many people are not aware. And then I think an important take-home message is also that there are science and technology innovations that can drastically enhance the scope and increase the speed and the precision of radiation breeding. And the final point is that in order to bring radiation breeding to the next, I would say the next level, that really investments in R&D and partnerships especially are critical to ensure that there's continued success for technology development and applications in the fields. So I think I stop here. Thank you. Yeah. Here's the team. I've not mentioned anybody in name, but I'd like to acknowledge everybody. And I mentioned some of the work that's been done over the past few years. Thank you.
Thank you, Dr. Ingelbrecht. That was a fascinating presentation. And I know it's very hard to get all of this information in such a short period of time. And we really needed to get the background on what the IAEA does and what the technology does, so that, and how we understand it, so we can better experience and understand the next speakers. I'm going to ask the speakers, though, to try and really keep to 12 minutes. I'm going to ask, please, also people sending in questions, do keep them to the question and answer button and not to the chat. But let us know if you have any issues on the chat. Without further ado, let's move now to Dr. Gomez. Dr. Gomez is an agriculture engineer and a senior lecturer in the crop science department of the Faculty of Agronomy of La Molina National Agrarian University in Peru. She has dedicated herself to collecting, preserving, and using germplasm from native Peruvian crops such as quinoa and kiwicha, and from introduced cereals such as wheat, barley, oats, and others in improvement programs to develop improved cultivars for the small farmers of the highlands of Peru. She has authored and co-authored publications of scientific works in the areas of cereals and native grains, and has received numerous awards for her research, including awards from Innovation in Agriculture, awards from UNESCO and from the Peruvian government, and a prize for the contribution to food security and sustainable development of agriculture from the FAO IAEA division. Dr. Gomez.
Good morning from Peru. Greetings to all the participants to the webinar. Thanks to the organizer for inviting me to present the research work carried out at La Molina Agraria University in Peru on the use of atomic energy in the genetic improvement of important crops for food and the economy of family farming in the Andean region. The challenges facing farmers in Peru can be summarized in the following presentation. Agricultural production takes place between 3,000 to 4,000 meters above sea level. Climate is characterized by frequent periods of drought, floods, torrential rains, frost, hailstorm, and other negative factors that make agriculture production unsafe. Plots generally smaller than 1 hectare. Most small plots are located on slopes in shallow, stony, and poor soils. Crops grow only during the rainfall season without any irrigation system. Family subsistence depends on few crops, among them potatoes, barley, wheat, taro, pea, faba bean, quinoa, amaranth, fruits, and some Andean tubers. 83% of the rural population survive in extreme poverty situation. Almost 23% of the total national population. Also, we have malnutrition problem, especially in children population at the rural area. From land preparation to harvesting, the farms use traditional cropping system with intensive use of hand labor, with or without the help of animal traction, because the major part of the farming community is still live under ancestral custom. They use limited agriculture impacts like quality seed, fertilizer, pesticide, and agriculture machinery, because poverty, difficult topography, adverse environment. Almost all the production is used for self-consumption. The farming technology applied is suboptimal because of poor knowledge, poverty, or the economic risk of farming in marginal area, and a very climatic negative effect. Frequently, the yield and quality of the crops are very low, and sometimes field yield only is enough for family consumption, which creates problems like malnutrition and poverty. There is a need to improve the yield and quality of crops in the highlands of Peru. Cereal seeds are widely accepted by the farmers because of the low cost and the easier introduction within the agriculture system. For this reason, the National Agrarian University started to work improving barley, wheat, and native grain. Our main objectives is to develop new cultivar for the small farmer with better yield, better quality, improve resistance to disease, improve tolerance to adverse environment condition, but able to grow with the farming technology, because it's very hard to change the farming technology because the poverty. So we start a program working, improving, making crop genetic improvement, agronomic technology improvement, and transference of results to the farmer. We use different types of methodology like hybridization and mutation induction, to develop varieties with the support of some biotechnology tools like double haploid production, molecular marker. Mutation can occur naturally, but take hundreds of years, and often the good mutations go unnoticed. The use of mutation induction accelerates plant chains, and all positive mutations can be selected for the creation of new varieties with high value. This helps to make agriculture profitable in rural areas in regions of extreme poverty. It is the low-income farmer and consumer in food deficit nations that have the greatest need to group improvement in negative environment in order to have food security. Using gamma ray, we developed 3 varieties that they are growing in the highland of Peru. Una LAN 95 variety was developed with the special purpose to replace varieties with naked grain that become susceptible to yellow rust in the Peruvian highland between 3,800 and 4,000 meters above sea level. Barley grain are roasted and is a special food for children during lunchtime in the school. Barley contributes with 20% of the caloric intake. It is the second source after potato, which contributes with 21% in the food of the Peruvian highland. Centenario mutant variety show 4 principal differences with the parental variety: better yield, early life cycle, better protein content, better test weight. The combination of these characters with a good plant height and resistant to yellow rust made Centenario very valuable for the highland farmers. It is the main barley variety cultivated in Peru. The mutant amaranth variety Centenario, with similar nutritive quality than the parent material, has better yield and increase equal to 29%, different plant color. At farmer location in the coast, the yield range from 3,500 to 5,500 kilograms per hectare, and in the highland from 2,500 to 3,700 kilograms per hectare. Centenario is today one of the most chosen variety, growing near to 47% of the cultivated area. It has high economic value, increasing the farmer profitability and reducing malnutrition problem. In investigation carried out to measure the impact of Centenario cultivar introduction to farmer production, and increase of yield was observed in Junín near to 50%, in Huancavelica 105%, in Huánuco 120%. Studying the profitability of barley production and increase in farmer earning of 180% for Junín, 193% for Huancavelica, and 163% for Huánuco were observed. Prices were slightly higher for Centenario due to the quality of the grains. The introduction of Centenario was made in more than 1,500 farmer communities, improving the yield, the consumption, and the profitability of farmer families. The introduction of the mutant varieties was made with the support of various institutions, such as non-governmental organizations, the Ministry of Agriculture, and other groups. At the university, we follow a very special protocol for the introduction of new varieties. based in one ancestral custom of farmers of the Andean region, which is the loan of seed among farmers. A bag is lent at the sowing time, and 2 bags are given back at the harvest time. In this way, it is possible to expand the growing area and increase the seed using the resource of the farmer. Amaranthus centenario variety was introduced to the farmer community by a company that processes and sells native grains inside and outside of Peru. In this way, farmers receive agriculture input for the crop season and have a safe market at harvest. By the use of improved barley cultivar with diverse methodology plus crop technology, farmers have a significant increment in barley productivity. Yield increase is near to 1,100 kilograms per hectare, considering the price per kilogram equal to $40 make $440 for hectare. One additional income for each growing season for the farmer. Centenario is adding $6,600,000 to the national economy yearly. Considering all 9 barley varieties developed by my university, the contribution to the national economy of the highlands of Peru is close to $18 million yearly. We have a new development. The work made during the last years include the improvement of highly nutritious crops such as quinoa. The use of this crop could improve the problem of malnutrition, especially of children, and can also improve the family economy, considering the high price of these crops at national and international markets. Quinoa advanced mutant lines were developed with better tolerance to high temperature and resistant to one important disease, downy mildew. Both are limiting factor for quinoa introduction to the Peruvian coast and other countries in the world. In barley, we have a new advanced mutant line with the better mineral and content for food and feed use. One mutant line with better amount of protein in the straw was selected in demonstration experiment and used as animal feed in the highland, improving the amount of milk production. Finally, in this opportunity, in the name of my university, the small farmers of Peruvian highland, I want to give my acknowledgement to the IAEA and to the Peruvian Energy Nuclear Institute for the support to the agriculture development in Peruvian highland through many coordinated research projects and technical cooperation projects. Maybe we cannot change the soils and the environment of the Peruvian highland, but we can change for better the socio-economic situation of the farmer with new and improved varieties using mutation breeding. Thank you.
Thank you so much. Thank you, Dr. Gomez. That was such a fantastic presentation and giving us such a valuable insight into the challenges facing farmers in Peru. And I'm struck by how mutation breeding has improved the livelihoods of the farmers and contributed to the economy in the country. I'm also very impressed with the collaboration between the research institutes, the government, and farmers. And I think your method of seed distributions to the farmers should be developed into a best practice guide. Honestly, thank you so much. And now we will move to our next speaker.
I welcome you all to my presentations, especially the moderator, and to the panelists, and to the participants from all around the world. My topic is the contribution of innovative nuclear technology to sustainable development in Bangladesh. I will go for some situation of Bangladesh. You know, Bangladesh is a very small country, and if you see to the global map, I think you will not see Bangladesh because it's a very small. It is occupying a very small area in the global map. You can see, but it has a very big population area, although it's 141,170 square kilometer, but population is more than 160 million. It is almost nearly half of US population, although the area is very small. The total cultivable area is 8.2 million hectares. For this, we have to grow even 4 crops in a year in the same land. And also, you know that our climate is much vulnerable. We have many problems to grow our crop around the year. One of the salient problem is salinity. You can see a salinity map here. Total saline area is 2.85 million hectares. And of the cultivable land, 1.05 million hectares is saline affected. And soil salinity ranges from 2 to more than 15 decicement per meter. And you can see this in this map, there are different types of categories of salinity, very high salinity to very low salinity. And this map, it is the middle part of the Bangladesh. You can see Dhaka, the capital. The salinity is coming from south to the north, and it is now at the middle. That means nearly the capital of Bangladesh, Dhaka. The salinity is coming. So, and in the dry periods of the year, the saline area, the salt deposition is like this. You can see the crust of salt here. This is cloudland. No crop can be grown there due to salinity. Only a single crop can be grown during the rainy season. When rains come, salinity goes down and farmers can't easily grow crop. In the other part of the year, they cannot grow any crop there. No crop can be grown during other times, and remain fallow during the dry period. And we have also drought problem. You can see the drought map, and the red is the highly severely drought prone area, and the pink is moderate. The pink color is severe, and the yellow color is moderately drought area. And you can see in the whole area there is some drought prone area. And we have also flood problem. You know, most of the year we have flood problem, and due to flood, the standing crop hampers. And in this slide, I am showing you the trend of minimum and maximum temperature. You can see the maximum temperature. The maximum temperature is in March, April, and the minimum temperature is in the December, January. And rainfall, you can see rainfall. We have mostly rainfall in the April to September. This is our mostly rainfall area. the remaining months has less rainfall and not sufficient to grow crop. Actually. And now I am coming to the mandate of the institute. You know, BINA, Bangladesh Institute of Nuclear Agriculture, is mandated to use nuclear technique for peaceful use, peaceful use in agriculture, and ensuring a stable and productive agriculture through evolution of new varieties of crops. That means breeding, mutation breeding, and scientific management of land and water, development of appropriate technology to improve quality and quantity of crops, and development of methods for control of disease and insect and pest management. So these are mandate, and already Ivan has shown very clearly the process of mutation breeding. So I will not go for that. As BINA is working on mutation breeding, now we have two sources, gamma sources, cobalt 60 sources. The left one is old. It was collected from Canada, and the right one is somewhat new. We have procured it 3 to 4 years back, but we are using both the irradiators still. The older one for lower doses, and the right one, new one, for higher doses we are using. And so far, using the mutation breeding, that means different techniques of mutation breeding, maybe gamma ray, maybe ion beam, maybe chemical mutation, maybe biochemical mutation, or maybe crossing between mutant and non-mutant. We have developed. Here I have shown 76, but recently we have developed some varieties, maybe 3 to 4, which was not listed here. But here you can see 76 varieties we have developed, and out of which 15 varieties from rice, 1 variety of wheat, and this variety is salinity tolerant, only saline tolerant variety in Bangladesh. We have 3 jute varieties. Bioseeds, we have 25 varieties. Pulses, we have 26 varieties. Tomato, we have 4 varieties. And spices, we have 2 varieties. And you can see from each graph the contribution of the different mutagen sources. Gamma irradiation has the highest contribution to our variety development. Then hybridization. Hybridization means a mutant, crossing hybridization between a mutant and a non-mutant. That is also a mutant. And we have also carbon ion beam irradiation. Chemical mutation we have. Biochemical mutation. Biochemical mutation means sometimes we use some extract of datura or some other plants to extract, and we use it for inducing mutation, and we were successful with datura. And we have 2 varieties developed, one from lentil and another from rice we have. And we have also chemical mutation and biochemical mutation combination.
Sorry, Dr. Azad, I'm going to ask you to move on to the 2 varieties that you want to discuss, as we're starting to run out of time. So you can skip forward.
Okay. I'm going faster.
No, yeah.
So far we have the traits we have attained using mutation breeding. One is, the most common is short duration, that means short lifespan in case of rice, or higher yield than the parent or the standard Czech variety in Bangladesh, improved quality in terms of protein, in terms of amylose, in terms of many things. Drought tolerance. We have drought tolerance varieties, rice varieties, lentil varieties. Salinity tolerance we have. In case of peanut, in case of wheat, we have salinity tolerance variety. High temperature tolerance. In case of rice, if the irrigated rice is transplanting, then the optimum time, the anthesis period coincides with the high temperature. And sometimes in Bangladesh, the temperature during the anthesis period goes up to 40 degrees centigrade. At that temperature, no rice can grow, fill their grain. That means becomes dry. But using ion beam irradiation, we have developed one variety that can even tolerate 40 degrees centigrade. No sterility is there. Low temperature escaping, and using the ion beam technique, we have already developed one variety that can escape low temperature. And we are now trying for low temperature tolerance in rice during the anthesis period. Another modification of biennial onion to annual type. We have modified this biennial onion to annual using the gamma ray. Highly photoperiod sensitive to insensitive. Many varieties we have in Bangladesh, especially in the rainy season, monsoon season, they are highly photoperiod sensitive. But when we use the mutation breeding, it has become photo insensitive. Lodging susceptible to tolerant. Many varieties we have. Less input variety, that means that they need less input to grow, less fertilizer, less irrigation. And finally, we have shortening the breeding cycle of rice from 10 years to 4 years. So you can see here, using the...
Dr. Azad, you have 2 minutes left, so perhaps...
Yes,
Okay, sorry.
This is the carbon ion beam irradiation we have used in our rice variety. And we developed these two varieties. You can see these two varieties. One is left and another is right. We started the program in 2009, and the variety we have released in 2013. That means only 4 years and 1 or 2 months needed it. Using the carbon ion beam irradiation, this is the most significant contribution, I think, of ion beam irradiation. And there are many beneficial sides of the ion beam irradiation is there. And we need much and more research on this, as it is not hazardous like gamma ray or other mutagen. But ion beam irradiation, I think this may be a very important mutagen for future mutation breeding program. So you can see in both the cases I need only 4 years to develop a variety from starting with the seed irradiation to release the variety. So I think I will show some the results. This is the variety Binadhan 14 wicket. It is high temperature tolerant, and it is highly. Its parent was, you can see here. Yes, its parent, no flowering, but the mutant matured. That means no photoperiod sensitivity in it. Grain size you can see. It can tolerate 13. It was developed by carbon ion beam irradiation, taking the help of Japan. And another, the same case, this another significant, I think, modification, a non-aromatic or non-fragrant rice I have irradiated with carbon ion beam irradiation. It has become aromatic. That means its grain has aroma. and its yield has been doubled, and its duration has been shortened by 15 days compared to its parent. And this is another variety developed by carbon ion beam irradiation, and it's drought tolerant. Its duration has been reduced by about 15 to 20 days, and this its parent was very staggered growing. Some plants are very high, some plants are very flowering earlier, some plants are mature earlier. So when we use the carbon ion beam irradiation, it has the uniform growth, and you can see the uniform growth, and it has drought tolerant character, and its duration lifespan has been reduced, and yield also increased. So
Dr.
I
Azad,
think,
could you take us to Binadhan 7 and just talk about the Monga, and then I think we'll wrap up.
This is another variety. It was its segregating material we collected from Vietnam, and this has a miracle effect in Bangladesh to reduce poverty. In the northern part of Bangladesh, every year they have shortage of food, fodder, and work. But introduction of this short duration variety in that area, the starvation has been over, and there is no poverty is there. Okay, and also some impact. I am interesting to show you. Yeah, you can. Sorry, with the introduction of this Binadhan 7, the area under pulses, oilseeds, vegetables has been increased, because due to its short duration, it accommodates the crop in the cropping system in every year in the same land, where 2 crops can be grown before introduction of Binadhan 7. Now they can grow, the farmers can grow 3 crops or even 4 crops with the introduction of the Binadhan 7. And I think, and I am interesting to show you the area coverage. The only variety we have, you can see 461,385 hectares of lands were grown last year. And I think in terms of money, if we have the cumulative, maybe hundreds of millions of dollars contributed this Binadhan 7 to our economy. So
thank you so much. I think we're going to have to end there.
Yeah, and I acknowledge to Mrs. Ingrid and also to Shobha, the head of the plant breeding division of IAEA, and all you who are connecting, joining, and listening my lecture. So thank you all.
Thank you so much, Dr. Azad. Actually, I mean, we could really spend an hour on this. Your work is absolutely fascinating, and anyone who is interested can go onto our website at the VCDNP and find a case study that we authored with the help of Dr. Azad, which goes into detail about all of the impact of these various plants and the work he has done.
Sorry, Ingrid, I forgot it. Yes, it is in your website, and also I have posted in my website also.
Fantastic.
So it was a very interesting challenge.
Fantastic.
So, absolutely fantastic.
It's really, the work is inspirational. Thank you so much. This is a very good segue now into Professor Tomoko's work, because up to now we've been talking mostly about plant mutation breeding that has been performed by gamma radiation through cobalt-60. And here in Bangladesh, as we've seen, there has been great strides made with the use of ion beam accelerators with a Japanese, in collaboration with a Japanese laboratory. Professor Tomoko.
Thank you. Thank you. I'm happy to introduce my research in this meeting. Today's my talk is application research using ion beams. Here shows new varieties developed in various crops using ion beam in Japan. The target of ion beam breeding have extended from flower to crops, and 70 varieties have been produced by ion beam breeding. Useful varieties have been grown, such as low browning cut lettuce, high yield lettuce, tearless and non-pungent onion, Pansenocarby tomato, late flowering edible chrysanthemum, short clam Japanese millet, low cadmium uptake rice, and good sake flavor and fermentability yeast. Sake bred with new one Saitama G yeast is sold as Nishina homare. あれで、次はどうするんだあれちょっと待って。これか。これか。あれおい、see. The user in RIKEN, which promote ion beam breeding, consists of more than 200 parties. There are many mutant lines that are expected to contribute to solving the global food and environmental programs. Semi-dwarf mutant isolated in rice, soybean, duckweed, and bell pepper. Duckweed mutant did not fall even in the strong wind of typhoon. Marine crops obtain good growth mutants. Larger mutants were obtained with animal plankton, rotifer. They are expected to improve the survival rate of fish baby and increase aquaculture productivity. High oil content mutants were isolated in chlorella and euglena. In Artemisia annua, mutant with high content or yield of artemisinin, a medical component with anti-malarial activity, were isolated. In peanuts, there are mutants with reduced proteins that cause allergies. Salt tolerance rice grow normally and produce good quality grain in saline paddy field. We have identified the responsible gene for salt tolerance mutant. In order to bloom the cherry blossom, low temperature are required to break the dormancy. Due to global warming, the low temperature will be insufficient and cherry blooms will not bloom. Shinsei is a new variety that do not need cold to break dormancy. This is the newest variety created by RIKEN and Shizuoka Prefecture. In Japan, the breeding of Satsuma mandarin is very popular, and agricultural experiment station in each prefecture is working on it. Due to global warming, citrus unshu bloomed early. Storing for a long time caused spoilage. The S1200 mutant flowered a month later than Aoshima unshu. During the storage of the fruit in room temperature, the citric acid content of the fruit decreases and the sugar content increases. Less likely spoilage due to less lindo puffing. Farmers are able to ship high quality fruits in March and April, when market demand has been growing in recent years. Gamma ray and X-ray can change only the dose. Ion beam can change LET in addition to dose. LET is linear energy transfer, which means the amount of energy given to the target per unit length. Gamma ray and X-ray are low LET radiation, and ion beam is high LET radiation. In general, higher LET means larger biological effects. We analyzed mutations in genome-wide in rice using some mutants. Large dilution are characteristic of mutations induced by ion beam. Large mutations have a tendency to increase the percentage with increasing LET. A LET of 30 kiloelectron-volt per micrometer was found to be the most effective of mutation in rice. This treatment shows high mutation rate, same as chemical mutagen. Number of mutated genes are 4 to 10, relatively small, and often only one mutation is found near the mapped location. It is easy to identify mutated gene related to one's phenotype. This table shows the novel genes identified using mutant induced by ion beam. Many new genes have been isolated from various plants. Based on yield test for 2 years, we observed 8 to 60% yield increase in the argon 576 plants when compared with that of the wild type. As the mutant plants had long reign, we performed full genome sequencing and linkage analysis to identify the responsible gene given the name long grain 1, LIN1. LIN1 can be a new target gene for higher yield for genome editing. Mutants have become more and more useful and important in modern genetic studies. The discovery of genes using the combination of mutants and genome sequencing technology may lead to the new field in biology, mutagenomics. Advantage of fast ion beam for mutation breeding in RIKEN. Irradiation treatment can be given to various plant materials smaller than 9 cm in diameter. High mutation rate and wide variety of mutants can be taken even at low dose. Short irradiation time, seconds to a few minutes. Less harm to the plants and damage fewer genes, thus requiring less time for breeding. Discovering novel genes using mutants. New genes can be targeted for genome editing. We build a new beam line to increase arrivable ions with higher LET. We can use argon ions with 184 kiloelectron-volt per micrometer for plant breeding. We will develop a unique technology for mutation induction using these heavier ion beams. We intend to spread the unique technology globally through international collaborations. Thank you for attention.
Thank you so much, Dr. Tomoko. Really amazing work that you've been doing, and I think especially as we move forward and there are more and more challenges with the impact of climate change, the work you are doing is going to help us to solve many of these problems or contribute hugely to solving them. Of course, ion beam accelerators are not within the scope or reach of the average country, and definitely not most developing countries. But exactly this example of how Bangladesh has worked so closely with Japan, and how Japan has supported food security and crop development in Bangladesh is an example of our regional cooperation and how countries, developing countries, can benefit greatly from those countries in their region that have these technologies. I'm sure we will hear much more about your work in future, and it's a great pleasure to have some insight into what you have been doing. And now we'll cross to Dr. Pillai.
Okay, perfect. So thank you for your patience. So this is my talk. I'm going to talk a little bit about electron beam and X-ray technologies. We have heard so far about the tremendous applications of gamma radiation and ion beams, but I want to use my few minutes remaining to talk about electron beam and X-ray technologies, which I think are extremely important from a sustainability standpoint. And also I want to give you my perspective of gamma irradiation technologies in terms of agricultural sustainability, and especially around the world, and then talk a little bit about the recent developments in E-beam and X-ray technologies. I think it's extremely important for countries around the world, especially in the underdeveloped and developing parts of the world, to realize what's available today off the shelf, and then show you some very specific scientific evidence for the use of E-beam and X-ray technologies for plant mutation breeding. And then finally, I want to end up with showing how Texas A&M University is doing our little share of trying to provide support to the IAEA and also to promote agricultural sustainability around the world. So if you look at nuclear technologies, I don't need to tell you much about it. Nuclear technologies, be it gamma or E-beam or X-ray, has done tremendous impacts at both the pre-harvest level as well as the post-harvest level. Pre-harvest, developing new plant varieties, surface sterilization of seeds, soil treatments, and also for sterile insect technology, animal vaccines, storage, pest controls, and controlling storage pests and long-term storage of grains, and also for food irradiation and increasing shelf life and improving quality. So it goes without saying that what the International Atomic Energy Agency has done in terms of atoms for peace and development, it makes absolute sense that this is a tremendous technology. So now the question comes. I know there are people around the world at this webinar, and the question is, what do I use? We have seen tremendous application of gamma irradiation, and then now we're talking about X-ray. We had the presentation from Japan about ion beam. So the question is, which do you choose? And I think I want to walk you through my thought process on this. And I think it's very important that at the end of the day, we talk about sustainability holistically. So the reality of cobalt-60 today, whether it's a gamma cell or a big cobalt irradiation facility, is that cobalt-60 availability will only decrease no matter what you're told. Do not fall for the premise saying that cobalt-60 is long-lived. Except in maybe one country or two countries, cobalt-60 is definitely limited. So you have to be very careful about what you're going into if you're going to the crop mutation program. Second is that cobalt-60 will become significantly more expensive and will soon become economically unsustainable. And this we are seeing in the United States as well, where cobalt-60 is becoming very, very expensive and becoming unsustainable. The ability to procure cobalt-60, the ability to transport it, to safeguard it, and other challenges will reduce your availability. So your access to cobalt-60 technology will be very limited. So the agricultural industry, and well as the scientists around the world, needs to become very knowledgeable in alternative technologies so wise decisions can be made quickly, because without that, you will be going down the wrong path. So in my opinion, this is a dead-end technology. I'm quite emphatic in my approach in this, but I really see what the trend is happening around the world. So my advice is to countries who are investing is, please do not go down the cobalt-60 route, because you're going to end up with a very expensive situation where you're not able to sustain it. So when you talk about technologies, ionizing radiation technologies, you have panoramic processing, gamma facilities that has been the workhorse for many, many years. You also have the electron beam facilities where you can treat these products in thousands of pounds per hour. And so this technology, whether it's gamma facility, electron beam facility, really is really matured. And then you really have to make a decision to go down the electron beam for panoramic processing. But when you talk about self-shielded chambers, when you're talking about for crop mutation breeding programs, everybody has heard about the gamma cell. They are a workhorse for many, many years, 50, 60 years. But you also need to be aware of the other technologies that are becoming available. For example, you have machines from X-ray machines that are treating, that can be used to create the same mutations. And these are significantly less expensive to maintain, procure, maintain, etc., than other technologies. You also have new companies coming in like PCT E-beam with X-ray system that can treat over 100 kilograms of material per hour. You also have Mavex, it's a Canadian company, has called Beams in a Box, which has both 1 MeV electron beam and 1.9 MeV X-ray capabilities in a self-shielded unit. And then you have a commercial company out in Switzerland that is doing this for spicy radiation. So this is now electron beam and X-ray technology self-shielded. So it's really plug and play. You don't have to worry about the cobalt-60 source in your facility. So this is important. The other important is that for those who are not familiar, and I pardon if this may be a little too, rudimentary or basic for those who are very well versed in this, in electron beam technology, and be aware this is not ion beam, this is electron beam technology. This is you use high voltage, electrons are accelerated to almost the speed of light, 99.99% the speed of light, and you get a very high beam of high energy electrons coming out. It's a very high dose rate, 3 kilograms per second. So you can know when you're talking about mutation breeding, which is in terms of grays, the significant value of this technology is very high throughput, extremely efficient and cost effective. But when you try to package the material for seeds, etc., you do have to spend some time. The other approach is using X-ray. But in this case, rather than using X-ray tubes, you use the accelerator technology, and then you use a tantalum shield to convert electron beams into X-ray photons. The only difference between electron beam and X-ray photon is that the dose rate is a little bit lower than, well, it is lower than electron beam. It is much higher, the throughput is lower, and maybe not as cost effective as electron beam. But what you get in these two technologies is highly adjustable. You can control the energy, you can control the dose rate, etc. And so this has tremendous value if you're trying out different energy parameters for your mutation breeding program. So these are the commercially available technologies, and I want you to become very familiar with some of the acronyms. If you look at the electron beam energy or the X-ray energy on the x-axis and the beam power or the power, LEAB is called low energy electron beam technology. LEAX is low energy X-ray technologies. And these are the technologies that are currently available that you can purchase. You do not need the type of, it is nothing more than the permitting process similar to an X-ray cabinet. So you do not have radioactive materials on your site. Any research lab, any academic institution, any company can purchase this equipment. You also have the medium energy and medium energy X-rays, which are what is called the first prototype is what we're getting at A&M is at beams in a box. And then you have the high energy electron beam and high energy X-ray. This is for panoramic treatment. And this is really where companies can come in and treat hundreds and thousands of pounds of food per hour, or not thousands of hundreds, at least millions of pounds of food per year, etc. So this is very high throughput system. But so what I'm trying to emphasize here is that this technology is commercially available, and you need to be able to make these decisions very carefully as to what you want. So the scientific evidence for machine sources of crop mutation. So do keep in mind that, as what is also mentioned, X-rays have already started showing a tremendous value. The number of mutant cultivars that's been released. This is from the FAO in 2015. About 910 varieties from gamma, 311 from X-rays. And surprisingly, if you look at chemical treatment, they are significantly less. Also from a treatment, from a chemical treatment system, is that even the treatment process takes about 3 to 5 days to achieve the mutation, while in X-rays and E-beam, you're talking about a few minutes to get the treatment done. Also, when you talk about ion beams, they are you need cyclotrons and very expensive. The cost of that is upwards of $35, $40, $50 million to get the ion beam capabilities in developing countries. So you do need to be aware of that. Also, when I looked at the literature that about the scientific evidence for electron beam for crop mutation, there are a number of papers starting back in the early 80s, as well as the more recent papers, which show that electron beam, not only X-ray, but now electron beam, can actually have tremendous value for crop mutation programs. In fact, it has a very high mutation frequency, about 2.5 times greater than gamma radiation. So this is very, very important to keep in mind. So the arguments supporting electron beam and X-ray technologies, this is what I want to mention. There are a lot of countries in this list that can be classified as developing emerging economies. They already have E-beam and X-ray technologies available. Pakistan, Mexico, Costa Rica, Brazil, Egypt. They already have this technology available. So it is actually very simple for the agricultural scientists or the mutation breeders to actually work in this area, because you can now have very controlled dosing, very high dose uniformity. The dose rate can be controlled. And more importantly, it's relatively low capital cost and operating cost. And this ultimately relates to technological sustainability. Now, there have been lots of arguments saying, well, there are some countries which you do not have enough power in the power grid, etc. But I want to mention there are countries like the Dominican Republic and elsewhere which are using generator sets to power big, very big electron beam facilities when the electricity grid is not dependable. So do keep in mind that all of the arguments you've heard about electron beam in terms of, oh, well, there's not enough power, that is absolutely not true. In fact, it's the fastest growing technology around the world. So what we do at Texas A&M University is that we have almost 19, 20 years of experience. Since 2002, we have been managing the National Center for Electron Beam Research. This is not a research, this is not a university lab. This is a very high commercial throughput facility. We have high energy electron beam. We also have the capability of X-ray technologies. And now more recently in a partnership with some of the federal agencies, we are being able to access low energy electron beam as well as low energy X-ray technologies. We have state-of-the-art dosimetry system. The facility is fully inspected, USDA FSIs, et cetera. So we do a lot of R&D work. We also do a lot of commercial work. So we can actually help countries to understand the business models to use this technology. We also have been offering hands-on workshops. Unfortunately, in 2020, we couldn't do that because of COVID, but we host a lot of visiting scholars, and through IAEA and other fellowships, we train a number of students. So this is really what we try to lay out for the National Center, is really to train and to advance this technology, machine source technologies around the world. So what our goal is that we partner with the Pacific Northwest National Labs and the National Nuclear Security Administration's Office of Radiological Security. Our goal is just beam it. It's like the Nike, just do it. We just call it just beam it. The idea is that we want to harness electron beam technology as a platform technology. We don't look at this as, oh, it's a very sophisticated technology. We look at this as, it has to be no different than your iPhone technology, but the applications are significant. We can make novel probiotics, novel biopolymers, vaccines, etc. The goal that we want to do, working with countries and companies, is to commercialize the technology. My opinion is that even after spending 30 years in academia, just publishing papers without really feeding the people makes no sense. So, without any commercial value. So our big goal is to innovate and to come up with commercial operations that enable companies to be started. More recently, we are working with Pacific Northwest National Labs and the NNSA, and we are getting the access to the low energy electron beam technology and low energy X-ray technology. So I want to make this invitation to all of you who are on this call. Please contact us. Probably in about 12 months, we should have both of these equipment available to us, and you're more than welcome to come up and spend some time with us to do the research so that you can go back to your countries and adopt these technologies. So what we do as an IAEA collaborating center is we harness E-beam technology for cleaning, healing, feeding, and shaping this world and beyond. We use it for environmental remediation applications. We use it for novel therapeutics, for feeding, food security, and food safety. So this whole discussion about crop mutation fits into that. We work with a number of companies to develop new bioplastics and novel polymers. And then we work with NASA and some of the private space companies to develop food for the space tourism, as well as for the NASA astronaut program. So that's our, as an IAEA collaborating center, is what we do. So we don't do this by ourselves. I have to, there are a lot of people around this call. I know from private companies, for example, Industrial and Avanti Mediclear. We actually work very closely with universities around the world. We work with the equipment providers, international agencies, international financing institutions like the World Bank, the Inter-American Development Bank. We work with government agencies and private investors because ultimately we want this technology to show up as economic improvement in the country. So we work with companies like eFoods and Avanti Mediclear, eAgro, and we come up with novel packaging systems, even coming up with a business model is how do we build out large storage facilities so that you can preserve food from spoilage, because you can imagine in a lot of the developing parts of the world, a lot of the food is destroyed even before it goes into distribution. So our goal is to have a holistic solution to this approach, starting from crop mutation, food processing, as well as storage. So I want to give you here a case study of how we have partnered with Mexico and Pakistan.
Sorry, Dr. Pillai, can you just, after this slide, wrap up? Thank you.
I will. Yeah. Thank you, Ingrid. So what we do here is for case study. So Mexico has been exporting a lot of the crops, a lot of the produce to the United States, and for a number of years, and it's gone significant development. So we partnered with eFoods Imports and eAgro Industrial, both out of Mexico, who are involved in using this technology for foods. And then we basically developed a supply chain all the way from Mexico. They come into Texas A&M for the mangoes being treated with electron beam. It gets treated in these boxes, and then these are mangoes even after 6 months of storage in refrigerated conditions. So we've really solved basically a supply chain issue and an exporting challenge for this particular commodity from Mexico. Also, this program was so successful that the local city actually celebrated this sort of an export program. So we are really going down to the very, very important issue of making an impact. The other issue is that with Pakistan is right across the globe from us, but they were very interested in exporting mangoes to the United States. So we developed a supply chain which involved shipping mangoes from Karachi to Houston via airlines, and then from Houston to Texas A&M University. And these are the quality of mangoes that we are treating that have come out from Pakistan. And what is also important, ladies and gentlemen, is that we are treating mangoes at Texas within 30 to 40 hours of harvest in Pakistan. So we are talking about we are talking about really an optimized supply chain process. So this is the impact of capacity building. And I take a lot of pride in some of the partners we have done. The work that we have done at Texas A&M University has led to the formation of 2 new E-beam X-ray facilities in Mexico. One is already in operation called Avanti Mediclear. It's an electron beam facility in Tijuana on the US-Mexico border. The other one is going to be commissioned probably in about 4 to 5 months called eAgro Industrial. It's an E-beam X-ray facility. What we have done overnight by this is now Mexico has a capacity for a crop mutation breeding program based on E-beam and X-ray technologies. So this is really where we want to make the impact. Is, we want to bring in capacity building. And in Mexico and in Pakistan, we enable Pakistan to show that the mango exports could grow fast. And right after that, Pakistan built its first electron beam facility in Karachi. Automatically now Pakistan has a capacity for a crop mutation breeding program based on E-beam technology. So what I want to emphasize here is that we really want to make sure that countries are equipped with the right technologies so that they become sustainable. So the take-home message, this is my final slide, is that self-shielded electron beam and X-ray technologies are now commercially available. Plant mutation breeding programs can now be harnessed by self-shielded E-beam and X-ray technology. So once you acquire this technology, you don't need to go and depend on other countries to have access to the technologies. And I sincerely believe that E-beam and X-ray technologies are key to sustainable agriculture around the world. So with my final slide, this is the way you can get hold of me, and I'll be more than happy to start a conversation with you about acquiring this technology and moving away from gamma sources to alternative technologies. Thank you.
Thank you so much.
Thank you.
Thank you, Dr. Pillai. This is the most comprehensive presentation in my experience that I've had on alternative technologies, and the help that and support that you provide at Texas A&M is really valuable. great to know about it, and I hope that there are people on this call who will really be able to make use of your support, especially also in Africa. So we've got some minutes left for questions, and I'm going to quickly. One of the questions that I have, I'm going to ask Ivan. It's from Amber Morgan, and she's asking, is there a need that the IAEA is seeing in the field that is not able to address, with its current resources, or think that it would be helpful for member states to step in with bilateral support? Ivan?
Yes, that's a big question. Can you repeat it? Is this the question about additional support or
yes, can I just ask Dr. Pillai, what's happening? Noah, why am we only seeing Dr. Pillai at the moment? Or is that just me?
It might just be you, but...
oh, there we go. Okay,
yeah, there we go.
No, that's fine. Okay, so the question is, yes, that's the one about what support the IAEA would need from member states in terms of bilateral support to be able to improve its support to countries in this field.
Yeah. I think that the take-home messages or lessons from this very interesting webinar is that there's a need for alternative technologies. We heard the machine sources, that there's opportunities there. And I think if we want to continue to use plant mutation breeding successfully in the next, 20, 30, 40 years, then we need to harness these innovations, really. We heard about X-rays. I think we've done some research, but there's a lot going on, but not really in depth. That's what I'm seeing, that there's not a lot of in-depth research going. It needs to, we need more fundamental knowledge on the kinds of mutations that are being induced, and investments, contributions, whether it's financial or in kind, partnerships, collaborations. Exactly what I think the last panel speaker said. This is important. Yeah, new technologies, resources, alternative sources, more knowledge on what actually is happening, so we can make the random process more specific, and there's tremendous opportunities for that.
And you, as the IAEA, in your laboratory, do you have all the technology that you need, or is there more work that you can do? You have an X-ray there, but as I understand it, it's not an X-ray that is yet adapted to, or you would like to adapt it.
Very recently, we acquired a new X-ray machine, a new gamma cell, in fact. And what's coming is exactly the machine that was described by the last speaker, combined X-ray E-beam. That is prepared. It's on its way to be shipped, but we haven't really used it. So I think the adoption of new technologies, whether it's machine source or advanced science and technology innovations for improving the mutation breeding process itself, this can be vastly accelerated, and that would help.
Okay, fantastic.
So I think we are talking now for the laboratory. What we need is, I think, partnerships with the kinds of members that we just heard about, and then closer involvement of the member states. There's a wide variety of member states, so that's why I'm, depending on the capacities of these member states, the needs might be different.
Exactly.
But I think it's a fascinating webinar, real eye opener. So very, very pleased to be part of it and to hear also the other speakers.
Fantastic. Thank you so much. And just a quick one more, not even a minute. Let's see, there was a question about what lessons would you like to pass on to other countries hoping to access the benefits of peaceful uses of nuclear technologies to pursue their sustainable development objectives. Can I ask Dr. Gomez, do you have a minute as a couple of seconds just to wrap up and to say what you would recommend other countries?
I think it's very important to work a lot of institution with the farmer. So anything that we can develop in our lab, in our experimental field, I think it's better to be tested by the farmer, because they will select what is better for them. I think, and also to have the market, it's very important. And network of people working with the same objective.
Fantastic. Thank you so much. And Dr. Azad, a short input from you. Oh, you're on mute. You're still on mute. There we go.
Thank you, Ingrid. This was a very nice webinar, I think, and we have we have learned many things from this webinar. One request I had to you through Tomoko, as she is using the ion beam technology in his experiments and in his research, and I am also I was also using this technology, but unfortunately I have no lost this connection with Japan. May I request you to you, Tomoko, to make any collaboration between her lab and my lab to continue this work. This is my earnest request to you. Thank you.
Professor Abe, would you like to say a couple of words just in closing? Yes, I can hear you.
I am happy to collaborate with Dr. Abel. So I hope to collaboration research with you, with him.
Fantastic, fantastic. No, thank you, and I want to just thank all of the participants, you and panelists. This has been a really interesting discussion. You have the contact details of Dr. Pillai, and we will also circulate the details of the panelists and participants that have agreed to have their details circulated. So please feel free to contact the people that you feel that you could get information from, or have additional questions, or send us additional questions that you may have. Thank you to all of you for participating. Thank you for your presentations. This is really a really important work, and as we move forward, I hope that the IAEA and all the member states will be able to support countries, the least developed and developing countries specifically, to really be able to increase their capacity to develop crops that are resistant to drought, resistant to all of the impact of environmental impacts of climate change, and that as Dr. Pillai has mentioned as well, that we continue to have and accelerate access to technology, because we are really at risk of losing that technology if we only focus on one type of technology, and there are many options available, and the more demand there is, the faster this technology will develop. So thank you so much for participating. Thank you to all our participants from across the globe. And we will be having on the 12th of May, there will be another webinar where we'll be focusing on the use of nuclear technology for industrial purposes, food safety, etc. So we look forward to having the participants back here again, and anyone who's interested to listening. Thank you so much for today, and have a lovely weekend and Easter.
Thank you.
Goodbye.
Bye.
Thank you so much.